Good morning. I'm Gary Ritchie here for Pharmaceutical Manufacturing Magazine. We're here in Chicago today at PittCon 2009. I have the opportunity this morning to speak with Dr. Neil Lewis,
Good morning, Gary.
Technical Director of Malvern. Neil, coming out to Chicago, I understand you're introducing some new solutions in the area of the PAT Quality by Design initiative.
Correct.
In the past 2 years, that's been the central theme for pharmaceutical applications. However, as some of the industry is moving toward more biomanufacturing, what kind of things do you see on the horizon for solutions in that area?
Yeah, well, I think that's right, Gary. The industry has talked, I think, a lot over the last couple of years about the issues that pertain to the particularly the small molecule QBD PAT issues, and I think with the emergence of the biopharmaceutical industry, the solutions are going to be different, and the technologies are going to be different. We're talking about aqueous media. We're talking about things that aren't necessarily compliant with near infrared, as an example, and not only that, I think the problems with large molecules relative to small molecules, if the industry has struggled with small molecules, I think it's going to struggle a lot more with large molecules, with proteins that are obviously a lot more complicated, and the issues that can be seen in the manufacturing industry they're going to be a lot more complicated, I think, with the biopharmaceuticals.
Larger molecules do pose a different set of problems for the manufacturers. For instance, what areas do you see that Malvern offers to solve some of those problems?
We have several new products that we've launched over the last 6 months or so. We have a couple of systems here that actually do dynamic light scattering, and they're specifically targeted towards the biopharmaceutical industry, so small volumes and looking at proteins and other kinds of biomolecules. So that's what these instruments do. This is a 96-well plate reader for doing things like formulation studies and that kind of thing, and this is a small volume system. And they do particle size, and they do zeta potential of materials in the nano-size scale range, so proteins, liposomes, drug delivery systems, those kinds of problems.
So this one offers a solution in terms of, let's say, screening or development.
That's correct.
And this one, once you have a method in place, I suppose you can do routine analysis.
Correct. This one actually looks at very small volumes, down to a couple of microliters of material, because again, there's not a lot of material available. Again, unlike the small molecule business, there's typically very small amounts of material available during formulation development.
Well, Neil, this is all very interesting. As we go forward in the future, the work that you're doing with Malvern seems to be taking off in different areas. You started out in the imaging area. Now you're getting into protein characterization. What do you imagine in the future coming out of Malvern and your research lab?
Well, I think there's that a need. Again, I believe that there's a measurement gap here for the biopharmaceutical industry that needs to be filled. I think this is one solution where dynamic light scattering can help address things like protein aggregation, issues that may lead to problems with release of a product. While right now we target in this at formulation development, stability test, and this, that, and the other, I think there's probably a potential for this technology, sometime down the road as a release test, and possibly even an at-line technology right in the manufacturing of biopharmaceuticals.
Well, that's great. Thanks again for speaking with me today.
Thanks, Gary.
And we look forward to hearing more from you in the future.
Appreciate it. Thank you.